gram-negative cocci. Up to now, we know about streptococcus and staphylococcus, including enterococcus. They are gram-positive cocci. Here they are gram-negative cocci. So mainly there are two types of organisms: Neisseria species and Moraxella species. There are other, but here we mainly concern about Neisseria and Moraxella. So what is Neisseria? Neisseria species, they are gram-negative cocci that usually occurs in pairs. Here they are gram-negative diplococci. Streptococcus, it is gram-positive diplococci. But arrangement per se also differ. Neisseria compared to Streptococcus is a chain, so it goes with the main axis. So it can seen as pairs in a line like, but in Neisseria they replicate horizontal 90 degree to Streptococcus axis. So it seems as pairs like two parallel pairs. So like kidney appearance. Neisseria species, Neisseria gonorrhoeae and Neisseria meningitis are pathogenic for human and typically found associated with or inside the polymorphonuclear cells, that is neutrophils. So term pathogenic Neisseria, always this intracellular or association or nearby appearance of neutrophils are very much important. Considering Neisseria meningitis, it can be a part of our normal flora or common cells in upper respiratory tract. But Neisseria gonorrhoeae, it's not a common cell. It's a true pathogen. But asymptomatically, the bacteria can be there in female genital tract. So gonococci and meningococci, the use for the sake of easiness, we say gonorrhea as gonococci and meningitis as meningococci are usually closely related. So there are 70% DNA homology and are differentiated by a few laboratory tests and specific characteristics. So Neisseria meningitis or meningococci have a polysaccharide capsule, but gonococci do not. And also meningococci rarely having a plasmid, but most gonococci they are having plasmids. That's two differences. So about the morphology and identification. Typical organisms. The typical Neisseria is a gram-negative, non-motile Diplococcus, approximately 0.8 micrometer in diameter. Individual cocci are kidney bean shaped, just like kidney bean shaped. The organisms occur in pairs. The plateau concave sites are adjacent. That's how they appear. So you can see here, these are the nuclear materials. So polymononuclear leukocyte or pus cell. And within neutrophil, later pus cell, they deny neutrophils and term pus cells. So gonorrhea is there within. Culture. So in 48 hours on enriched media, it requires enriched media. So we say these organisms are fastidious. Compared to Staphylococcus, Streptococcus, they require additional nutrition like hemine, other factors. So we have to provide enriched media. So once they grow in a culture, it appears as convex, glycerin elevated mucoid. So colonies in 1 to 5 millimeter in diameter. Colonies are transparent or opaque, non-pigmented and non-hemolytic. I think now you are familiar with the hemolysis. So gamma hemolysis, so we'll say non-hemolytic. Further, Neisseria grow best under aerobic conditions, but some grow in anaerobic environment as well. They have complex growth requirements, what I said. And most Neisseria oxidize carbohydrates, producing acid but no gas. That means when they produce gas, it's a fermentation. Here, oxidize. So, oxidize or positive oxidase reaction is there. Oxidase is positive. What is oxidase test? Detect in the presence of terminal enzyme system in aerobic respiration called cytochrome C oxidase or cytochrome A3. So, for the oxidation, they require cytochrome C, cytochrome oxidase enzyme system. So when it is present, oxidase test invariably become positive. So procedure, briefly, take a filter paper, soak with the substrate, tetramethyl phenyl adenamine dihydrochloride. So that is the oxidase substitute. Moist the paper with the sterile distilled water. So moisture. So because it's soaked with this oxidase reagent. Pick the colony to be tested with wooden or platinum loop and smear the filter paper. So in here, to take wooden or platinum loop, if we take iron rod, it having a tendency to oxidize. So it gives false positive results. So that tries to eliminate that error. We use wooden or platinum loop. Observe inoculated area of the filter paper for color change. So once this is the strip, dip in oxidase reagent, and once add some moist or drop in with the pressure pipette or just a pipette tip. Then when you take the colonies, suspecting colonies using the wooden or platinum loop. So then once you apply here, within short period of time, 10 to 30 seconds, it gives this color change. That is turned into purple color. So it is oxidase positive. So other important thing is here oxidase negative. That means no color change. Always we have to compare with the positive control and negative control. So the positive control is Pseudomonas aeruginosa. Negative control is Enterobacteriaceae coliform. You will get familiar with these things. Further about the growth characteristic, both Gonococci and meningococci grow based on media contain, I said, complex organic substance, that is from heated blood, heme, and animal proteins, and the temperature requirement is, and also atmosphere, that is 37, and atmosphere contain 5% carbon dioxide. Growth is inhibited by some toxin substances in the medium. Fatty acids, salts are there. Other thing is the organism is killed rapidly by drying, sunlight, moist heat, and many disinfectants. So that is about the prevention or infection prevention part. But at the same time, in here our main idea is make the isolate, get the growth of the isolate. This is a fastidious organism. So we have to provide enriched media, heated blood, other proteins. Then we have to provide the atmospheric temperature as well as 5% CO2. And also we have to eliminate these toxin constituents to enhance the growth. We'll bit about Neisseria gonorrhoeae. I said it is oxidase positive and it only oxidizes glucose only. So it's a test we use to differentiate from other gonorrhoeae. So here gonococci oxidize only glucose. That's the thing. And it usually produce small colonies compared to other gonorrhoeae.